I'd like to welcome you to today's Dairy XNet webinar series. Today's webinar on potential and pitfalls of genetic selection is our topic today. First of all, I'd like to go ahead and introduce myself. I'm Donna Amaral Phillips, Extension Dairy Specialist at the University of Kentucky and the project leader for Dairy XNet. So I'd like to go ahead and introduce today's speaker. Dr. Chad Dechow is an Associate Professor of Dairy Cattle Genetics with a teaching and research appointment at Penn State University. He co-advises the Penn State Dairy Club, Dairy Science Club, and coaches the Pennsylvania 4-H dairy cattle judging team. He grew up on a small dairy farm in southwest New York, and he joined the faculty at Penn State in 2003. He has an associate degree in agricultural science from SUNY at Mooresville, a bachelor's in animal science at Cornell University, his master's from Penn State in animal science, and he earned his PhD in animal science from the University of Tennessee. The primary focus of his research program is improving dairy cattle health and well-being while breeding for higher levels of milk production efficiency. And with that, I invite Chad Dechow to talk to us about the genetic selection and the importance of that. Thank you, Chad.
Okay. All right. Thank you very much, Donna, for that introduction. And I want to thank everybody for joining us here today to talk about genomic selection a little bit. And I want to start out by saying that, everybody has a little bit different perspective on genomics and where the right and wrong areas to use genomics might be. So you're going to get my perspective today, but understand that not everybody would have the same outlook I would. But I'll try to walk through where I think it is most useful as we go along here. And so some of the basic things that I'm going to cover is I'm going to start by looking at a little bit about a review of the genomic technology in the 4-path genetic selection model, which is what motivates geneticists to get excited about things like genomics. And then we'll look at, we'll compare early genomic predictions to actual daughter proofs to give you a sense of how accurate and how much confidence should I put in this, and maybe more importantly, how can I spread risk and who should be using and maybe who should not be using genomics. I will spend a little bit of time on talking about genomics as a herd management tool. And then if we have time, we'll get into inbreeding a little bit and then beyond SNPs. I thought I'd start out here by looking at one particular gene that has a big effect on dairy production traits. And that gene is called DGAT1 for short. And it's an enzyme involved in triglyceride synthesis on chromosome 14. And when we genetically engineer a mouse to knock that gene out, we get a mouse that has complete absence of milk production. So that's one of the ways we were able to determine that this gene is involved in milk-related traits. And what it turns out is there is a binucleotide substitution. So our genome, and I'm assuming that we're going to have varying levels of background when it comes to genetics and genomics in the group. So if I'm covering stuff that seems obvious to you, I apologize for that. But we have about 3 billion nucleotides in our genome, and same thing for cows. And in one particular spot, there is a change from an AA to a CG, just one small little mutation. And what that does is it changes a lysine amino acid to an alanine amino acid. And when we make that transition, we end up producing about 300 pounds more milk, 5 pounds more protein, but it comes at the expense of fat production. And this should be a negative 0.17% fat and negative 13 pounds of fat. And in addition to that, once we make that transition, we produce a little less saturated, a little less saturated fat and more non-saturated fat. And so that's a pretty big effect. That's terrific. But this cow over here is the world record holder Jersey for fat production, and her record is over 2,800 pounds of fat. So that if she has 2 copies of this lysine variant, that only explains 26 pounds of her fat production, out of 2,800 pounds. So So that's terrific, but obviously we need a lot more information than that. And that was, we started out by looking for these types of genes and pretty quickly realized that that wasn't going to get us that far. So we have transitioned to a whole genome approach, and where we look at single nucleotide polymorphisms. And basically a single nucleotide polymorphism, or SNP, is just a one base pair change in DNA. We estimate that there are 10 to 50 million present in the genome. I've seen various estimates. However, they're not inherited independently of each other. And when we get to inbreeding, I've got a slide that'll maybe help you visualize why that is as we go along. But basically, cows have 30 chromosomes, and one chromosome is essentially one long strand of DNA with many genes on that one single strand of DNA. So our genes are not broken apart into separate segments. We've got one gene that's lined up right next to the other gene and so forth. So there's a few different tests that we can use to determine some of these single nucleotide polymorphisms. The one that forms most of the basis of our genomic predictions currently is the bovine SNP50, which tests for 50,000 SNPs throughout the genome. There is also a somewhat cheaper option where we can called a low-density chip, where we can test currently 9,000 for about $45. And then because a chromosome is one long strand of DNA, we can use that low-density chip, and we're actually able to make some very strong, educated guesses about the remaining 50,000. And so we can do that with over 99% accuracy. And then there's also a newer option called a high-density chip, which tests close to 800,000 SNPs. The early research has not been very exciting about that chip in terms of it adding any value beyond the 50K chip at this point. Okay, now there is another option as well. We could do some whole genome sequencing, and this is just an example of one bull that had his genome fully sequenced. The reality is that we are a long ways away from being able to use all of that information. Just in terms of the amount of data that's generated is overwhelming, and we can't really analyze that data in an efficient manner. So whether that actually ever becomes commonplace or not, I don't know. I tend to think that'll just be for research purposes as we go along. But that option is potentially available in time. This slide here is from USDA, and what it is showing us is the effect of those 50,000 SNPs across each of these chromosomes. And this particular slide shows the effects for fat yield. So this is chromosome 14 that I've put the pointer on here in the black. And you can see that chromosome 14 has one effect that explains about 14% of one additive genetic standard deviation for fat yield. And that effect is, of course, DGAT, and that is by far the largest effect for this particular trait. And then there are a couple other spikes, if you will, that might explain 2 or 3%. But the main point is here, that one line looks pretty tall relative to the rest. But if we start stacking all of the rest of these lines on top of each other, that would give us our total genetic potential, and you can see that would really dwarf this one line. So that just shows why we need a whole genome approach when we're evaluating our genetic prediction. Okay, and then this is just the same thing for final score. The difference here is that you can see there really isn't any one single large effect that explains a lot of genetic variation for a cow's classification score. It's really just many thousands of genes that each one has a really small effect, but cumulatively there's a very large effect. Okay, so this formula here tells you why geneticists get excited about genomics. And this formula tells us how much genetic progress we are going to make in 1 year's time. And there's really only 4 functions that determine how much progress we're going to make. One is how accurate are the PTAs that we're using when we do our genetic selection. And in the dairy industry, we use the term called reliability to measure PTAs. Second is selection intensity. And selection intensity is simply a function of how picky we are when we decide which bulls we're going to use. So for example, we can use the top 10% of sires to breed to our herd, the top 1% of sires, or the top 80% of sires. And the pickier we are, the higher the selection intensity. The next formula is simply the amount of genetic variation that we have in a population, and that's something that we have very little control over. A population either has variance for a trait or it doesn't. And there are some traits that are genetically determined, but that have no variance. So for example, if we wanted to select for cows that had 5 legs, the number of legs on a cow is determined by genetics 100%, but there's no variation for it, so we won't make genetic progress towards selecting for 5 legs. And then the last component that we have here is the generation interval, which is just how old our parents are when we're born. Okay, so we can take this formula and we can break it down a little bit into the sire and dam pathways, and then further into the sire of sire, dam of sire, sire of dam, and dam of dam pathways. And essentially this sire pathway, this is happening at the level of the bull stud. And obviously we have very intense selection in terms of the bull studs are only using the very best bulls in the world to create their next generation of bulls. And they're mating them to some of the very best cows in the world. So we have very high selection intensity. The challenge that we've always had is that in order to find out who the very good bulls are, we had to wait until a bull was about 6 years old before he had daughters that were giving milk. So that meant that our generation interval here at the bottom was very high, which we don't want a high number down here. So that slowed genetic progress. We also had, for the dam of sire pathway, very low accuracy because a heifer has no offspring. A cow might have 1 or 2 offspring. So really low accuracy of their genetic evaluations. So genomics has helped to increase that as well. So really, this genomics is helping us in 3 different ways. First, and the most important by far, is that it's lowering our generation interval. Secondly, we've got higher accuracy for females. And then we can make some progress on the selection intensity side because now we don't have to go through the expense of progeny testing a bull. We can just do a genomic test and we can test more bulls indirectly that way. I do want to caution you a little bit with that formula. We make some assumptions with that formula that aren't strictly speaking true, and some people overestimate the amount of genetic progress we'll make because of it. But in general, it's a good guideline for how we can make genetic progress in a population. Now these genomic proofs, which are testing 50,000 SNPs along the genome, were implemented officially for the first time in January of 2009. And it's been adopted pretty quickly. So these are the number of matings made to young sires in dairy herds in the U.S. And you can see we were below 30% in 2008. And in 2011, in Holsteins, we were approaching 50%. And I expect that now more than 50% of cows are mated to young sires. And by young sire, I mean a bull that has no daughters yet. Okay, and the bull studs are using these young bulls, not exclusively, but for the vast majority of their selection decisions. And how bull studs market this information varies a little bit. Some bull studs will have a separate lineup where they have bulls with daughters in one group and bulls without daughters as another group. And then some bull studs just mix them all together and they have all their bulls on the same page. So what I want to do is I want to make a comparison of, in January of 2009, when we first had genomic evaluations, I want to look at those bulls that had no daughters and look at what their genomic PTA suggested they would be, compared to now that they actually have daughters, now in 2012, what is their daughter deviation for milk yield and productive life? And I'm basing this on 517 bulls that had no daughters in 2009 and that have at least 100 bulls now. There are some additional bulls that would qualify, but I also put in an additional qualifier that they had to have daughters that were relatively old in order to make accurate evaluations of how productive life genomic proofs have been doing. And you can see here for milk yield, we've done pretty well. We have an R-squared of 0.55. So in general, we're doing okay in terms of our genomic predictions for milk yield. There are some exceptions. You've got a bull down here that was supposed to do pretty well, and he's now about average for milk yield. So there are some hits and misses for sure. Here's a bull that turned out to be a little bit better than we anticipated. Okay, so it's not as accurate as a progeny test by any stretch, but it does rank the bulls with reasonable accuracy. Productive life is the trait that has caused the most problems for us, and you can see the accuracy of productive life is quite a bit less, only about 34%. And there's a fairly large pocket of bulls down here that were supposed to be good for productive life and that have turned out to be average or even a little bit below average for productive life now that they actually have daughters. And we'll talk a little bit more about this as we go along, but certainly productive life is one of the traits where we have to be a little bit careful with our genomic predictions. So we can take these R-squared numbers and then adjust those for a couple of different factors to derive what I'll call a realized reliability. And so basically we have to adjust for the fact that the bulls that had a really low genomic proof are not going to have any daughters because no farmer is going to want to use a bull that is supposed to be terrible. So after we've accounted for that, the realized reliability for milk yield in Holsteins was about 69%, and we had predicted that it was going to be more in the mid-70s. So a little bit of a drop from what we expected, but not too bad. Daughter pregnancy rate was in the general range of what we expected. The trait that was lower was productive life. We expected this to be more 65%, and when we do the calculations with the actual bulls, we get more 55% or so. So, the proofs have been fairly accurate, much better than what we've had in the past for young sires, but still not at the level of what we would expect from a progeny test. Perhaps what surprised me the most when we look back at those bulls is when you take the top bulls in 2009, okay, that had no daughters. So this average for 2009, this blue column here, were the top genomic young sires in 2009. And you can see that their net merit, PTA, was almost $700. When we compared that to the top proven bulls in 2009, those proven bulls were the top 25 only averaged 500 daughters. So we had almost a 200-pound jump, and when you looked at that, you'd be a little bit crazy not to use the genomic young sires. But fast forward to 2012, where we now have daughters available for those genomic young sires. And I've only included proven bulls in here that added at least 50 daughters to their proof. Now those top 25 young sires and proven bulls are essentially the same. So it didn't matter in 2009 whether you used the best young sires or the best proven bulls. The actual genetic merit of those bulls, the true genetic merit, was about the same. However, the very best, the elite bulls, were those that were the genomic young sires. So if you were into marketing cattle and you're trying to develop elite animals, then yeah, genomic young sires made sense. If you were just a commercial milk producer, then you could get away without using genomic young sires and still be making about the same amount of progress as you would have otherwise. And this is just a chart that shows a few bulls of how they've changed, a few popular bulls The one that people talk about is Freddie, because he, in August of 2008, before we had genomics, was about plus 500 pounds, which was still pretty good. But then when we did his genotype, we recognized that he should be an exceptional bull. And in fact, as he's added daughters, he's still the top in the breed, so he's done very well. But there are some other bulls that we forget about. Casino is one of those, and he's dropped steadily over time. Scholten is another bull that's dropped a little bit. However, he dropped more because net merit is a combination of several different traits, including the amount of fat and protein yield in productive life also has a very big influence on net merit in daughter pregnancy rate. When we revised the formula in January of 2010, it went against him. It wasn't in his favor, and so he got a little bit of a drop there. So he dropped, but less so because of his genomic predictions for different traits and more because the formula changed. Atwood is one of the most popular young sires that's been used. I recognize the reason that Atwood was popular is because his daughters have very high classification scores. They look really good, they win at the shows and so forth. And in fact, now that he actually has daughters, they are in fact doing that. And so from that standpoint, they've held up well. But his genomic proof, particularly for productive life, was quite a bit overestimated. And now that he has daughters, he's dropped a little bit as well. So there are lots of success stories and then a few cases where things haven't gone quite as accurately as we'd hoped. So the traits that we need to watch for most intensely are productive life and calving-related traits. In particular, calving ease has not been projected as accurately as we'd hoped, and that's a real problem when you're breeding your animals to virgin heifers. If you're basing that on a calving ease evaluation that turns out to not be accurate, you can run into some troubles, and there are a couple bulls that are examples of that happening. The other trait, going back to productive life for a minute, one of the reasons that productive life has been problematic is because the reasons that farmers cull cows change over time. And we're making our predictions based on past generations, and we put a little bit of emphasis on bulls that had good udder composite. We assumed they'd have daughters that would live a little bit longer. If they had getting good feet and leg composite, we assumed that they'd have daughters that would live a little bit longer. And what we've done now that we've realized that the genomic predictions for productive life haven't been as good is we've gone back and we've re-estimated some of these things. And we found out that farmers aren't putting as much weight on productive life. Feet and legs isn't having as big of influence as it used to have. On the other hand, daughter pregnancy rate is having a greater influence than it used to have. And somatic cell score is having a greater influence than it used to have, as this number is stronger than it was in the past. And there's more negative selection pressure against body size than there has been in the past. So the scientists at USDA are hoping that that will help to improve the stability of productive life proofs over time. But again, only time will tell if that turns out to actually be the case. So who should use genomic young sires? Certainly those involved with marketing need to use them. There'll be hits and misses, and that kind of goes with the territory. Those people that are not marketing cattle, just commercial milk producers, they can certainly use genomic young sires. They need to watch the calving traits on their virgin heifers, and they need to spread their risk by using a selection of genomic young sires. They shouldn't be using 100 units of bull A because they really like his proof. They need to be using a selection of many bulls and a little bit of many bulls to spread their risk. And they need to be willing to accept the fact that there are going to be some misses. Those producers that aren't marketing and that want to minimize calving interval, that want to minimize difficult calving issues and that are willing to miss out on the best bull for 3 years probably shouldn't use genomic young sires. And I would point out when we looked at those top 25 bulls, the average of the proven bulls turned out to be the same as the average of the genomic young sires. So they're not going to be falling behind their competition if they decide to wait and use proven bulls versus young sires. Okay, I want to look beyond sire selection here just a little bit and to think about how we might be able to use genomics as a herd management tool by genomically testing some of our cows. And some of these slides aren't as important, and in the interest of time, I won't spend a lot of time on them. I just highlight a couple tools that are there for you as we go along. One thing that I think we can do is probably, we used to evaluate cows, and for example, we might look at this cow and say, well, she's got a lot of set to her hock. I'm going to breed her to a bull that will straighten that leg out a little bit. Setting aside whether that's a good idea to begin with or not, I think that we can probably move away from that visual appraisal type of selection and look more at that cow's genomic proof. I'm also going to take you to a couple web pages. We can't link out to the web pages, but I've got some screenshots of a couple web pages that look at chromosome PTAs. And the only people that would be interested in this are people that are doing really heavy marketing. So if that's your interest, I've given you a website at USDA that might help you with that a little bit. And I'm not going to spend a lot of time on this. But basically what we can do is we can start to look at a cow's predicted transmitting ability on a chromosome by chromosome basis. So this is Freddie, who is the #1 bull with daughters in his proof in the U.S. And we can look and see chromosome by chromosome. He does have, even though he's the best bull, he's got a few chromosomes. Chromosome 5, chromosome 20, chromosome 26 that aren't ideal. And so when we're mating Freddie, we might look for a cow that we think can improve these particular chromosomes. Okay, but that's trying to breed the elite bull to market for artificial insemination purposes. There have also been some research. There has been some research that looks at what is the theoretical limit for selection for milk production, and what could our top PTAs look like. And you can see here for our Holsteins, it depends on how you make your assumptions about recombination and so forth. But in theory, we could get PTAs up over 25,000 pounds of milk for Holsteins, and potentially as high as 75,000 pounds of milk. And actually, those would be breeding values, which would be twice the PTA. But you get the point that we are not anywhere near exhausting our genetic potential for milk yield. And the same is also true for fertility. So daughter pregnancy rate, there's still a lot of genetic variation left in our populations to improve daughter pregnancy rate. Okay, I'm going to skip this slide here in the interest of time, other than to say that producers can start to genotype their own bulls here shortly. And some of those producers may decide that they want to market their own bull directly rather than selling them to the bull stud. Okay, we're going to spend a few minutes talking about genomics as a herd management tool. And the premise here is that genomics can play a role for commercial milk producers, and particularly those with heifers to sell. And the scientist that's done the most work in this area is Dr. Albert DeVries from Florida. And I've given you a link to some of his work. I don't have time to go through every point that he makes in some of his work. But I've given you this to hopefully help you think about these things a little bit. Okay, and the basis for this is that because of sex semen, we've really seen an increase in our heifer replacement rate, or our potential heifer replacement rate. And I'm out here in Pennsylvania, so I just looked at for New York and Pennsylvania, and we used to have about 80 heifers for every 100 cows. And then in 2011, primarily because of sex semen, we were up over 110 heifers for every 100 cows. So what are you going to do with all those heifers? Well, one of the strategies has been to simply increase our culling rate and get rid of some of our older cows that were broken down and replace them with a younger animal. And of course, some herds are also growing internally and growing their herd size. But I'm going to just assume here for a minute we want to maintain herd size, and we've got all these sex semen heifers. What are we going to do? And there's a couple options. One's to increase our cull rate, which will help us we'll have fewer problem cows, older cows with mastitis problems and so forth. On the flip side of that, it means that we've also got milking a few less cows that are mature, and that should be giving a little bit more milk for us. Okay, and the other problem is that we still have to raise those heifers and feed those heifers. So it's not helping us to lower our feed costs, really. So another option is to begin to look at selling those heifers, and then the question becomes, which heifers do I want to sell? And that's where genomics can help us out a little bit and help us to improve our herd. So what Dr. DeVries has done is asked a little bit about, okay, what is the value of testing if I want to sell some of my bottom end ones? So I want to keep the top 70% of my heifers, the top 80%, or the top 90%. So what happens to the value of the calves that I keep when I do this? And I will make a point that before we do genomic testing, we want to our first culling threshold should probably be to get rid of our sick and diseased calves. And if we're doing that and we're only getting rid of the bottom 10% of our calves, then genomic testing probably isn't all that important for us as a herd management tool. However, if we're going to get rid of the bottom 30% of our calves, then it can become a lot more valuable. So this is what happens to the net merit of the calves that you keep if you are doing genomic testing. And this is comparing if I don't test any calves and I just get rid of, in this green line, I'm going to get rid of 30% of them. And I'm just going to do it mainly based on guesswork. Versus, on the other hand, if I test all of my calves and I get rid of 30% of them, the remaining 70% will have a gain of $200 worth of net merit. So I've made, by using that genomic test, I've improved the value of the calves that I keep. Okay, if I test 50% of them and still sell the bottom 30% of my herd, then it's more like $100. Okay, now that's great, but the test isn't free. So Dr. DeVries has also adjusted the value of this by taking out the test costs. And when you do that, your remaining calves should have an economic value of about $130 more than what it would be if I had culled them by just guesswork. Okay, now this assumes that we know nothing at all about the parent of those calves. However, we should know who the sire of that calf is, and we might very well know the dam of that calf and her genetic merit as well. So what really would be a more ideal situation is to rank your calves based on the genetic merit of their sires or their parent average if you know the genetic merit of their dam as well. And so Dr. DeVries has accounted for that as well. And what he has done is he's determined what percentage of the calves do we need to actually test to get this advantage. And so he's making the assumption that, and it's a correct assumption, that if I have a really good sire for one of my calves, I probably don't need to be doing genomic testing on that particular calf because the probability of her being in the top, the probability of that calf being in the bottom 30% or bottom 10% of my calf population is pretty low. So I don't need to be testing the top half of my animals. And likewise, the ones that are the really poor sires, I probably don't need to test them either because the chance of them being a really good one and me accidentally getting rid of a really good calf isn't all that great either. So what he's done is he's looked at, okay, what gives me the highest net present value? And essentially, if I'm keeping 70% of my calves and I'm able to pre-rank on both the sire's genetic merit and the dam's genetic merit, so the parent average, then I don't need to test the top half of my calves, and I don't need to test the bottom 10% of my calves. So from the 50th to the 90th percentile, I'll test some of those animals, and then I'll be able to get rid of those that need to be sold without spending quite as much on the testing. Okay. He also has done some simulations based on if you know only the sire and you don't know the dam, 90% of calves being kept versus 80% versus 70%. So he's got, but I don't have time to go through each one of those scenarios for you. And really, it's the general point that's important, and not the exact numbers themselves anyway. One of the challenges we may run into in the future is if we have a lot of people selling a lot of heifers, our marketing potential for those heifers might not be as great. So by selling them, we're still saving on feed costs. But another option may be to consider using beef sires on the bottom end of our herd. So this was, in concept, it's very similar to what we just went through, except for rather than determining which calves I'm going to sell, I am now going to determine which cows I am going to breed to beef sires. And that's an area that still needs more development and work in terms of developing numbers, hard numbers like I had in the last few slides. But it's another general idea for you to think about in terms of using genomics to help manage your herd. Okay, I've got one slide here. I think in the future we may be able to manage a cow according to her genotype. So for instance, if you've got a cow with a really low yield potential, she's a candidate to try to get bred back as quickly as you can and to keep the generation interval a little bit shorter for that cow. Cows that have really high dairy form, we know that they're just genetically designed to dump every ounce of energy they've got, whether it's from their feed or from their back, into the milk pail. And they are at really high risk of metabolic problems due to high body condition score loss. And there is a nutritionist from the UK that believes such cows should only calve with a body condition score of 2.75 to 3, which sounds pretty radical to us here in the US. And I think it needs a lot more work to develop this idea, but it's just kind of throwing that out there as something that down the road we may want to consider is, can we manage our cows based on their genotype? Okay, so I've got a few minutes left here to talk about genomics and the rate of inbreeding. And I'm going to start here by showing you a slide from Paul Van Raden at USDA that I thought was just gives you a really neat depiction of what happens during the process of reproduction. And so this is the pedigree of a bull named Megastar. And you can see he was the result of a brother-sister mating. So Megabuck and Digny was the German cow, was his dam, were half siblings. Megabuck's sire was Aerostar. In Aerostar, that particular sperm that eventually became Megabuck, he had 2 copies of chromosome 24, 1 copy that he inherited from his sire, and 1 copy that he inherited from his mother. Okay, remember, a chromosome is just one long strand of DNA. And sometimes during meiosis or during reproduction, we get a crossover event and we'll mix the sire's DNA with the dam's DNA. And sometimes we don't. And in this particular case, there was no crossover. So Aerostar passed on his entire chromosome 24 to Megabuck. Okay, Megabuck's dam, you can see on the other hand, she passed on a chunk of her sire, and then there was a recombination event, and she passed on a chunk of her dam. Okay, Digny, the exact same thing happened. Aerostar passed on his entire chromosome 24 to his daughter with no recombination events. Her mother actually had a double recombination event, so she passed on a bit of her sire, a small amount of her dam, and then a little bit more of her sire. And then, just by random chance, Megabuck again passed on that entire chromosome 24 with no recombination. And Digny did have a small little recombination here at the event here at the end, but for the most part, passed on that entire chromosome as well. So what does this have to do with inbreeding? Well, inbreeding by definition is a percent of the genome that is identical by descent. And you can see here in Megastar this area where he has, from Aerostar, the exact 2 copies of the exact same chromosome. That is where Megastar is inbred along chromosome 24. So you can see he's almost entirely inbred on chromosome 24, except for that small little bit there at the end. Okay, so, and this is also why we don't need to test every single SNP, because if we know what SNP Megabuck inherited from Aerostar right here in the middle of the chromosome, we can make a very accurate guess about what SNP he inherited right here and right here and right here and so forth, because it's all one great big long linkage group. Sometimes we make mistakes, but not very often. So the bottom line is, I think we are going to accelerate inbreeding with genomics because we have a shorter generation interval. Secondly, the technology that we're using is what you might want to think of as pattern recognition. It recognizes a pattern of SNPs, and if you have an individual that has an unusual pattern, that wouldn't be recognized, and we'd call them average. We wouldn't recognize them as being genetically superior, because we wouldn't have any information to base that upon to realize that they really were good genetically. So what we are ending up doing with genomics is picking those that have the most recognizable patterns, or in other words, those that are most related to the breed. So that is also going to accelerate inbreeding in our populations. However, on the other hand, what genomics does is it gives us an opportunity to develop some inbred lines more quickly. This says live, it should say line. So we can develop our inbred lines a little bit as they do in the poultry and swine industry. And then these inbred lines can then be crossed in the commercial cow population to avoid inbreeding in the commercial cow population, even though the lines themselves might be a little bit inbred. So, there is some potential for genomics to help us on the inbreeding front if line development works, and there are some efforts to develop some of those. Or, on the other hand, if that doesn't work, it could paint us into an inbreeding corner. Last 2 minutes here, I just want to talk a little bit about, if we know the DNA code, why aren't genomic tests 100% accurate? And there are a few different reasons for that. And I think the main lesson that I want you to get from this is that we never know as much as we think we do. I'll show you that on the next slide as well. And part of this is that the markers are randomly selected. We don't necessarily test for the genes that are the most important. We just randomly select these markers and hope that they're pretty close to the important effect. The second thing is that there is copy number variation in terms of the number of genes that animals have. I have a colleague at Penn State who's looking at the copy number variation for genes on the Y chromosome, and he's found that some individuals might have 5 copies of a gene, the next individual might have 30 copies of a gene, and he's been able to relate that to the fertility of those bulls. We're also not accounting for dominance effects or interactions between genes, which I think are pretty important. We're not accounting for that yet. We will over time. And then lastly, we're not counting for what we can call epigenetic effects. And this is really kind of an exciting new area of genetic research. And what we've found is that we can have 2 individuals with the exact same DNA sequence, but there's an extra layer of control on top of that genome that determines what genes get expressed where and when. And so they might have the exact same DNA sequence, but express those genes differently and end up with slightly different phenotypic outcomes. Okay, and here's just another point. The more we learn, the less we know. There used to be something called pseudogenes, and we still call them pseudogenes, that were thought to be relics of evolution in non-functional junk. Just this past fall, the idea that all of those were non-functional junk has been pretty thoroughly debunked by the ENCODE project. And so now we know that the genome, maybe not all of it has a function, but more of it has a function than we've ever thought. And there's actually, in terms of dairy cattle breeding, there's a big effect for calving ease on chromosome 18 that localizes to a pseudogene. And for a while, we thought that something was messed up and that can't be correct because it's a pseudogene, which is not non-transcribed junk. Well, in fact, it probably is the effect of that particular pseudogene, now that we've found out that pseudogenes do have some function. So the bottom line is we have a long way to go before we get to 100% accuracy. So I've got a few take-home points that I want to emphasize, and then I'll be finished up here. And I apologize. I realized this morning that I didn't put a take-home message slide in here, so I apologize for that. But the first take-home message is this, that the genetic progress in the dairy industry is going to speed up because we have reasonably accurate proofs on young animals. Those proofs are not as accurate as progeny tests, okay, but industry-wide we will make faster genetic progress. Commercial producers, on the other hand, that are just milking cows and selling milk are not going to lose a lot of ground by waiting 3 years to know who the best bull is before they use them. And when they do use genomic young sires that don't have daughters, they need to be spreading their risk and not using a lot of semen from one individual bull. The next take-home point is that if you have excess heifers to sell or you want to mate some of your lower-end cows to beef sires, genomic testing does have a role to play and can help you make more informed decisions about which to sell or which to mate to a beef sire. But you don't need to test every animal. You probably don't need to worry about testing the top half of your herd based on their pedigree information. And you probably don't have to test the bottom 10% or so, but it's more the middle where you need to be doing your testing. So there's a lot of exciting possibilities with genomics, and hope you've learned something that you can take home and that will be useful to you. And with that, I'm happy to address any questions that we might have.
Thank you very much, Chad. I'll go ahead and start with the question, the first one that was in there, Chad. Does your testing strategy consider parentage accuracy?
Okay, so I'm assuming that relates to if you're deciding you're going to do genomic testing for your herd to decide which heifers to sell, and I said that you don't need to worry about genomically testing the top half of your calves if you know who their parents are and you know that they've got parents with good genetic potential. That assumes that you do know parentage accuracy reasonably well. If that is not the case, then you need to test a larger percentage of those. And maybe I'll pass that thought on to Albert. One more thing to add to a simulation is the accuracy of parentage in your population could, in most herds, about 15% of calves are sire misidentified. So there might be some loss of accuracy when you're doing that, and you might want to consider genomically testing a little bit more if that is the situation. So that's a good question.
I think this sort of goes along with that, but what about testing heifers not only for culling purposes but also for mating purposes?
So again, that's a question about can I do corrective mating, find out some of the better bulls to mate my heifer to? And my answer is that I think you'd be much better off spending the money on semen from a better bull. And that's, if you are interested in showing or marketing and so forth, then yes, you can find out genomically some of the areas where your animal could be improved and find some bulls that line up with her really nicely. But in general, I think we're better off just buying semen from the better bull rather than spending money on the genomic test.
Another question, can you explain realized reliabilities again?
Okay, so this is when bull proofs come out and you look in the bull book, there is a reliability attached to various numbers. Those reliabilities are a little bit of a guess. This is how accurate we think our proofs are. The realized reliability is taking a backward look to how accurate were those proofs in reality. Now that we actually have daughters milking from those bulls, how good did we really do? And those have tended to be a little bit less than the published reliabilities.
Okay, next question. How often have the genomics tests been used? How often have the genomics tests been used?
Currently, any bull that you purchase from one of the six larger bull studs are going to be genomically tested. So they're genomically testing all of their animals. I think probably this number is going to be a little bit rough, but maybe 40,000 bulls have been tested. I'd have to look at the most recent statistics, but that's my guess. There's actually more cows being tested than bulls now using the low-density chip. But all bulls are that you can buy commercially for AI are essentially genomically tested before they entered the progeny test program now.
Okay, Cheryl asked the question, have you evaluated any unselected, meaning they've tested all females of a certain age commercial herd data? In other words, have you worked with a commercial herd, I believe, to look at genomics?
I have. I've seen some of the reports that those herds get, and one herd did share their data with me. So the answer is yes. I don't know. Did I specifically evaluate it as much as just gave him my opinion about how he should use the information. But certainly commercial herds are genomically testing some of their animals in trying to figure out how to use it. So I'm not quite sure what she was driving at here, but that particular producer had, trying to sell several heifers and a fairly large percentage, and had tried to use that test. And I've talked to a couple other producers as well, and a lot of them were like, well, I did this because it sounded like a good idea, and I think it can be, but they weren't sure if they were utilizing it to its full potential, and they weren't convinced that was worth the full cost of what they were paying. And so that's a situation where I think you can target a certain percentage of your calves based on their genetic pre-ranking of their sires rather than all of them.
So what I'm hearing you saying is to wisely use your dollars then.
Yes.
Which animals that you test.
Yes. Yeah.
Okay. Les Hansen had a question. Are you concerned about the tendency to heavily use extremely high-ranking individual young bulls rather than groups of high-ranking young bulls?
Very much so. Yes. And that was the point that I thank Les for allowing me to hammer this point home one more time. I think especially for commercial producers, you need to be spreading your risk by using as many young bulls as you can. I think there's also an issue on the inbreeding from the population side of if the bull studs, there's that one bull that's really exceptional, there's going to be a tendency to want to really use that bull in all your contract matings and make the next great one. And that's been a problem even when we had progeny testing to accelerate the rate of inbreeding in some of our peer breeds. And that does concern me. It needs to be something that we as an industry maybe address a little bit differently. But certainly for a commercial producer, they should be using groups of bulls, use 5-10 brothers rather than the one elite animal.
So let me just follow up on that a little bit. So when you're talking about using a group of high-ranking bulls, you're talking 5 to 10 different bulls at a time in there, or do you want more than that, less than that?
It depends on the size of your herd. Okay, if you have a very large herd, several thousand cows, even more than that.
How about as a percent?
Ooh, that's a...
So you're talking a 100-cow herd using 5 to 10?
Yes.
So if you had 1,000, you'd multiply that by 10, or at least 5, so use 25 different bulls?
Yeah, probably 25 to 30 different bulls. Really want to... There comes a point where you use too many, you dilute. You're picking a little bit further down the list, and that's not helpful as well. So there isn't necessarily a hard number that I can give you, but that makes sense if we're 1,000-cow herd, maybe using 25 at a time rather than 5.
It doesn't seem like any more is typing. So I've got a question that you alluded to a little bit. It's a little off base from genomics, but you talked about there as far as the potential of dairy cows as far as production was concerned. Mm-hmm. And I always know that there's the environmental effects and then obviously the genetic effects. From a genetic standpoint, what kind of production genetically do our cows have? This is a broad question. I mean, double what we see,
Yeah, so that's a
1,000-pound cow isn't out of the realm. I'm just.
Yeah, that's so. I think that in general, most producers probably have higher genetic merit for yield under optimal environmental conditions, if you will, than what they actually realize. So I think most herds that have been using artificial insemination in their Holstein herds, and you've been using reasonably good AI sires, you haven't been selecting exclusively for type, for instance, but you've been using from the top of the AI list, you probably have the genetic potential in your herd to produce in excess of 30,000 pounds already. But not very many herds are there. So that tells me that we're not maximizing our genetic potential at this point. And I think that's part of the reason why some producers are shifting their selection focus a little bit toward fertility and some other traits, is that they realize my cows can give enough milk. I probably would have to change my milking frequency and some other things, build a new facility to get more milk out of them. So I'm going to focus on some other traits to help me out a little bit. So the answer is I don't think most herds are using their full genetic potential, but I also don't think that the genetic potential for 50,000 pound herds is sitting there unrealized either.
Well, I just thought it was interesting how you talked about the difference between there, because you've heard that for years.
Yeah.
Okay, another question. Should researchers be doing more to prop up evaluations of the unrelated genomically tested bulls to attempt to prevent driving the breeds towards selecting the lines we know the most about? Are you comfortable with line breeding techniques?
All right, so if we are, it depends a little bit what the purpose here is. If we are trying to develop separate lines, I am comfortable with doing that. And there is some effort now to keep lines that are completely separate. That's actually a good idea from an inbreeding standpoint. I think what the questioner is referring to is that our breeds in general have been essentially adopting a whole breed-wide line breeding where the breed genetic diversity keeps narrowing a little bit, and we've increased the inbreeding within a breed, not necessarily within a specific line within a breed. And that, I think, has a little bit of problems associated with it in terms of accelerating the breed-wide inbreeding level. However, we can develop lines that are pretty highly inbred, mate those lines separately in the commercial population, and have a commercial population with low inbreeding. So it depends a little bit what the angle of the questioner means there, I think.
Well, Chad, I think our time is up for today. We really thank you, and thank you for the very practical approach to how we use genomics, and thank you very much, Chad.
Well, thanks again for everybody tuning in.